Thank you very much, and good morning, everyone. My name is Florian Lempp. I'm a scientist at BayViro Biotechnology. We're located in the beautiful Mission Bay in San Francisco, and the mission of our company is really to develop novel therapeutics to fight global infectious diseases. Now, as our company is still pretty young, all the research data and all the research projects we're working on are still proprietary, and I'm not allowed to talk about those today. But instead, I'm very happy to talk about some of the work that I've done during my postdoc at the University Hospital in Heidelberg, Germany. So essentially, what I will do is I will give you a deep dive into the molecular virology of hepatitis delta virus and also into the development of novel cell culture system to study the viral infection. Chronic hepatitis delta virus infections are considered the most severe form of chronic viral hepatitis with the fastest progression towards liver cirrhosis and hepatocellular carcinoma. HDV is a satellite virus of hepatitis B virus, so it always occurs together with HBV infection, either in a co-infection or in a super-infection setting. And about 5% of all chronic HBV patients are co-infected with hepatitis delta virus. That leads to a total number of around 15 to 20 million patients chronically HDV infected. And just to set that into perspective, that's almost half of the amount of HIV infected patients worldwide. So it's actually a lot of delta virus infected patients. The good news is the HBV vaccination is also effective against hepatitis delta virus. However, for chronic HDV infected patients, there is no effective treatment available. PEG interferon alpha is used in some occasions, but the success rates are very low, about 10% cure rates. Now, we regard hepatitis delta virus as a neglected human pathogen because of different reasons. First of all, not many people don't know at all that HDV exists. So if you might just want to ask your neighbor if he has heard of hepatitis D or delta before, you might come up with very interesting outcomes. I'm curious what you can tell me then. The second is there is no treatment available. Like the treatment that is available for hepatitis B virus, like nucleoside analogs, doesn't affect hepatitis D replication at all. And together with that, also the testing rates, so the diagnostics of hepatitis D virus infections are extremely low. So in principle, every HBV chronic carrier should be tested once in their life if they are co-infected with hepatitis delta virus. But in reality, in a recent study from the US, only 8% of all HBV carriers have been tested for co-infection of hepatitis delta virus. So many of these patients don't even know they are chronically infected. The virus itself is a pretty interesting virus. It's a circular single-stranded RNA genome of negative polarity. It's very small, only around 1,600 nucleotides. It encodes only one open reading frame that can express two different proteins, the small and the large hepatitis delta antigen. Now, HDV requires the hepatitis B virus envelope proteins for virus assembly and secretion. That's why it relies on a co-infection with hepatitis B virus. So essentially, hepatitis delta virus hijacks the HBV infected cell and then envelopes its own RNA genome in the same envelope that HBV produces. So as you can see, HBV has a DNA genome, HDV has an RNA genome, but the envelope and the envelope proteins are exactly the same because they are from HBV. And because the envelopes are the same, also the cellular entry of both viruses is similar. Both viruses use the sodium taurocolate co-transporting polypeptide, or NTCP, as a cellular receptor. Now today I would like to talk about two topics. First is a more technical topic. I will show you how we have produced novel virus clones of all 8 delta virus genotypes that we can then use to do combinatorial envelopment with the different genotype HBs antigen proteins from HBV. And in the second topic, I will move deep into the cell culture models of hepatitis delta virus and show you how we developed a novel cell culture model that supports the complete HDV replication cycle. So for hepatitis delta virus, there are 8 different genotypes characterized with distinct geographical locations. So genotype 1 can be found worldwide. Genotype 3, for example, can be exclusively found in the Amazon region of South America. Genotypes 5, 6, 7, and 8 are located in Africa mostly. So although there are 8 different genotypes described, all the in vitro HDV research that has been done, and I need to point out also here HDV is neglected. There may be a handful to 2 handful labs worldwide actively working on hepatitis delta virus. And all of these labs for their studies use only one single virus isolate, which is the genotype 1 construct, first isolated by John Taylor in the 1980s. That's why I call it genotype 1T. Interestingly, that virus isolate is not even from an infected human, but it has been isolated from an experimentally infected woodchuck. And that woodchuck has been experimentally infected with the serum of an experimentally infected chimpanzee. And the chimpanzee has been infected with the serum from an infected human. So the sequence and the isolate of that genotype 1 construct is actually quite far away from a human sequence. And if we look at the other side, for the HBV side that we need for the envelopment, situation is not much better. There are 10 different genotypes for HBV described, but all of the research for hepatitis delta virus, all of the envelopment has been done only with one single genotype, which is HBV genotype D, that is also mostly used to study HBV infection in vitro. So all of the research so far has been done with genotype 1 delta, enveloped with genotype D HBV, although there are so many different genotypes available. But this is a clear lack of resources because the different genotypes for delta are just not available as molecular clones. So we wanted to overcome that and had the aim to generate and characterize cell culture clones for all of the 8 different HCV genotypes, and also to develop the tools like antibodies that recognize the produced delta antigens of all of these genotypes. And then use these genotypes to analyze HDV-HBV genotype combinatorial preferences. We started off taking sequences of the 8 different HCV genotypes just from databases. So we retrieved it from NCBI. In silico, we made a HCV 1.1 overlength antigenomic sequence, had the synthesized and cloned it into a pcDNA vector. For HBV, we have the genotypes A to H available in the lab, so we subcloned subgenomic fragments of those that encode only the HBV envelope proteins, because we only need those in plasmids that do not have a plasmid promoter. And for the tools, we had to develop a monoclonal antibody that is pan-genotypic and allows us to detect hepatitis delta antigen of all of the 8 genotypes. So we immunized mice with a purified delta antigen protein. We generated hybridoma, screened these by ELISA first, and all the positive clones we further screened by immunofluorescence against all of the 8 different genotypes. And indeed, we found 2 different clones, FD3A7 and FD5C4, that reacted against delta antigen of all of the 8 different genotypes in ELISA, in immunofluorescence, and also in Western blot. With these tools in hand, we could now start out and look at the replication of our newly established virus clones of the different genotypes. So for this, we transfected HH7 cells with the different virus clones. We took the lysate every second day and analyzed by Western blot at day 12 all the different lysates. I'll show you the results for the prototypical genotype 1 Taylor. What you see is that early in the replication of the virus, the small delta antigen is being produced, can be detected already at day 2. And then later in the viral life cycle, an RNA editing step occurs that allows also the formation and production of a large hepatitis delta antigen that starts at around day 4. If we do that with all of our newly developed virus clones, you can see that all of them start replication in cell culture, producing small and also large delta antigen. All of those except for genotype 5, where we don't see production of any delta antigen, indicating there is no replication going on. So this virus clone seems to have a defect. With that in hand, we could now start the combinational envelopment. So we wanted to have combination of virus produced of all of the 8 different delta genotypes, enveloped with the envelope proteins of all of the 8 different HBV genotypes. So it's a lot of combinations. We co-transfected the different plasmids in HH7 cells, took the supernatant at day 10 to 13, which contains the infectious virions that have been produced, and analyzed these in a reinfection on HH7 NTCP cells that we infected and fixed at day 6 and stained infected cells for hepatitis delta antigen. Now this slide is unfortunately pretty busy. You see on the left side, these are the different delta genotypes we looked at. And at the top, these are the different HBV genotypes for the envelope proteins we looked at. If we only look at genotype 1 of delta, enveloped with the different HBV genotypes, you can see that all of these produce infectious viruses. However, the total levels of envelopment are quite difficult. We see higher levels, for example, for B, D, and E, and lower levels for like A and C. If we look at the other delta genotypes, like 2, 3, and 4, we see generally less virus being produced. But also we see preferences for certain HBV envelopment genotypes. If we look at delta genotype 5, we don't see any infectious virus being produced, which is consistent with the Western blot data I showed you before. Interestingly, if we look at genotype 6, 7, and 8, here we see a clear preferences of delta envelopment with HBV genotypes B and also genotype E. So this is a huge increase in infectious virus being produced compared to other HBV envelopes. To sum up that part, we have developed 2 novel monoclonal antibodies that detect delta antigen of all HDV genotypes. All of our new HDV cell culture clones, except for the genotype 5 clone, replicated in cell culture. And we've enveloped these with proteins of 8 different HBV genotypes that were able to package HDV variants. And the HBV genotype B was best for envelopment. That's easy to remember. And the differential envelopment indicated preference for certain combinations, for example, HDV genotype 6, 7, and 8, enveloped with HBV B and E. With these tools now in hand, we can actually start the most interesting studies, which are going on now back in Heidelberg, which is to investigate the effect of these different cell culture clones on antiviral drugs that are currently used or currently in clinical development, like interferons or entry inhibitors, or infection and replication of all of the different combinations and all of the different delta virus genotypes. With that, I would like to switch to the second part of my talk and deep dive into the HDV cell culture models. So for HDV infection in vitro, what is currently used are cell lines, hepatoma cell lines that overexpress the viral receptor NTCP, like HH7 NTCP or HepG2 NTCP. These cell lines allow HDV entry into the cell and replication. The virus can replicate and produce hepatitis delta antigen that we can stain in the nucleus here. However, then the replication ends because all the next steps that include assembly and secretion of the virus require the HBV co-infection, which is not possible in these cell lines. So we don't have secretion of infectious progeny virus. We cannot infect any neighboring cells. Therefore, the aim of our study was to generate cells that support the full HDV replication cycle, including progeny virus secretion and reinfection, and then to use these cells to analyze antiviral effects of current HDV investigational drugs. We wanted to do that by overexpression of the HBV envelope protein stably in these cell lines, so that the virus can enter, replicate, secrete progeny that then can be used for reinfection experiments. We generated these cells starting from HepG2 cells that we transduced with the lentivirus to express the receptor NTCP, and then we transduced them in a second step with another lentivirus that has a specific cassette that encodes for the HBV envelope proteins under the authentic promoter without any plasmid promoter and the authentic viral polyase signal. Of these co-transduced cells, we isolated single-cell clones, and the best clone we named HepNB 2.7. We validated that HepNB 2.7 cells express the receptor NTCP, that they express HBV envelope proteins, large, middle, and small, and that these HBV envelope proteins are also secreted into the viral supernatant and can be detected by ELISA as HBs antigen. Now, for the most interesting experiment, of course, we wanted to know, do these cells now secrete progeny virus after we infect them and initiate replication of hepatitis delta virus? So for this experiment, we used HepNB 2.7 cells in parallel with HepG2 NTCP cells that do not express the HBV envelope proteins. We inoculated the cells and fixed them at day 40 post-inoculation, just to look at the initial infection. And as you can see, both cell lines can be infected with hepatitis delta virus, no matter if they contain the envelope proteins or not. Now, to see if there's progeny virus secretion, we took the supernatant from day 12 to 14 after infection and used the supernatant to inoculate naive HH7 NTCP cells and stained them at day 6, also for delta virus. Delta antigen, and what you see if we take the supernatant from the HepG2 NTCP cells in a secondary infection, we don't see any infected cells. This is what we expected. However, if we take the supernatant from the HepNB 2.7 cells for a secondary infection, we do get a secondary infection, so there is infectious progeny virus being released from these cells. So these HepNB 2.7 cells, this is really the very first cell line ever described that secretes progeny virus post-HDV, authentic HBV infection. We also did kinetics and looked at the long-term infection and secretion. So we see HBs antigen being secreted from these cells for more than 30 days, and also infectious progeny virus being secreted from these cells for more than 30 days. Interestingly, the total amount of infectious progeny virus directly correlates with the amount of inoculum that is being used for the initial infection. With these tools now in hand, we could start off investigating the current drugs that are developed in clinical phase 1 and 2 studies. And these are namely Mercledex-B, which is an entry inhibitor that targets the NTCP, thereby blocking viral entry into the cell. Lonafarnib is an assembly inhibitor which inhibits the farnesyl transferase. This is important because a prenylation step of the large delta antigen is needed for assembly of the virus. So with that, virus cannot be secreted into the supernatant anymore. And we also used interferon alpha that is already being used in the clinics with limited success. The experiments were designed in a way that we had heparin B2.7 cells that we inoculated with HDV, and we treated them with increasing concentrations of the different compounds throughout the study. We fixed the cells of the primary infection at day 12 and analyzed infection by in-cell ELISA, and we also took the supernatant from day 10 to day 12 in a reinfection experiment inoculating HH7 NTCP cells in a secondary infection, which we also looked at by in-cell ELISA. If we look at treatment with the entry inhibitor Merclodex B, you can see that in primary infection already we have a dose-dependent decrease of HDV infection up to 0% of infection. This fits very well to the described mechanism as an entry inhibitor, and the same holds true for the secondary infection. We see the same dose-dependent decrease. We can calculate an IC50 at around 1.4 nanomolar. We don't see any signs of cytotoxicity. If we look at the immune modulator interferon alpha, we also see a dose-dependent decrease in primary infection and secondary infection, however, not to 0, but there is still a remaining level of around 40% of initial infection, with an IC50 of 28 international units per milliliter. Also here we don't see much of a cytotoxic effect. Now what was interesting for us is when we looked at the prenylation inhibitor Lonafarnib. First of all, we looked at secondary infection because it's supposed to block virus secretion, and here for the first time we can show that this really is the case in an authentic infection model. So we see a dose-dependent decrease of secreted infectious virions and IC50 of 36 nanomolar. There is some cytotoxicity, however, at much higher concentrations. But what was very interesting for us is when we looked at the primary infection treated with Lonafarnib, because here what we did not expect, we actually saw an increase of hepatitis delta antigen in the cells that are being infected during primary infection. So this was weird. We did not expect that, and we wanted, of course, to get more insights into the mechanism behind that and where in the viral life cycle does this affect the virus actually. So we went into more detail, used again heparin B2.7 cells, inoculated them with HDV and treated with 50-fold DEC50 concentrations of the different compounds, and then looked by different methods at all the steps in the viral life cycle. So we checked by Western blot, immunofluorescence, and RT-qPCR. So first we checked by Western blot for the different forms of delta antigens, and as you can see, if we compare the untreated with the Lonafarnib treated, you can see that the levels of small hepatitis delta antigen remain the same. However, the levels of large delta antigen in the cell increase around 8-fold. Now just to repeat that, there are two different forms. The small delta antigen is required for replication of the virus. The large delta antigen actually inhibits its replication and is required for assembly. So with that result, we really wanted to know where in the cells or in which cells does this large delta antigen accumulate. So we developed a polyclonal antibody that is specific for the large hepatitis delta virus in a way that it only recognizes the C-terminal extension of the protein. And we used this antibody in immunofluorescence staining, which is shown here in green. Again, if you compare the untreated with the Lonafarnib treated, you can appreciate that, first of all, there is a lot more large delta antigen in the cells that were treated with the prenylation inhibitor, and also that the localization of the delta antigen is different. While it's localized only in the nucleus in the untreated cells, it bleeds out into the cytoplasm and accumulates in both nucleus and cytoplasm once you treat it with the prenylation inhibitor. We also looked at the intracellular levels of replication by assessing viral genomes by RT-qPCR. And as you can see, once we treat with Lonafarnib, we get an around 2-fold increase in total delta virus genomes in the cells. And it has been recently described that hepatitis delta virus infection is able to elicit an innate immune response in the infected cells, which is in sharp contrast to hepatitis B virus. So it's the co-virus that it co-infects with, which does not do that. So we looked at that by doing qPCR for interferon beta and interferon lambda. And as you can see, also in our heparin B2.7 cell model, when we infect with the virus compared to the uninfected, we get an increase of interferon beta and interferon lambda. And if we now treat with the prenylation inhibitor on top, we see a 30 to 40-fold increase of induction of innate immune response as measured by interferon beta and lambda. And also if we go down the interferon cascade and we look at interferon at ISGs like RS82, MX1, and IFID1, we can basically see the same. Once we treat with the prenylation inhibitor, we get a 10 to 30-fold increase in ISG production. So prenylation inhibition increased intracellular HDV genomes by 2-fold, which is accompanied by an enhanced innate immune induction by 11 to 40-fold. Now to sum up that part, we have generated the HepNB2.7 cell line, the first cell line that supports the complete replication cycle of hepatitis delta virus. These cells stably secrete infectious progeny virus for more than 30 days. We use these cell lines to evaluate antiviral drugs currently in clinical development and showed that nucleodex B completely blocks viral entry at an IC50 of 1.4 nanomolar. Interferon alpha reduced viral replication, but did not completely abrogate viral infection. Lonafarnib, the prenylation inhibitor, completely blocked HDV progeny virus secretion, but at the same time led to an accumulation of large delta antigen in the cells, as well as viral genome, thereby increasing the innate immune induction. And now we would like to propose our new cell model, the HepNB2.7 cells, as an ideal tool to first of all study the complete viral replication cycle, starting from viral entry up to assembly and secretion of progeny virus, to perform drug mode of action studies, as we have done it here in this example, but also to perform antiviral screening, like small molecule screening, but also screening for host factors, whether it's siRNA or CRISPR-Cas9. And it can also be used to support clinical studies, for example, to analyze or expand serum virus from patient serum, for example, from different genotypes, or also to analyze viral quasi-species. It can be used for clinical study monitoring to look at serum infectivity, because we can use patient serum from patients infected, directly put it on our cells and see infected cells. And we can do viral resistance testing with that. And with that, I would like to close and would like to thank my group back in Heidelberg in the Department of Molecular Virology, which is headed by Stefan Obern, and also my funding sources at the German Center for Infection Research. Before I open it up for questions, I would like to switch back into my role as a scientist at BayViro Biotechnology now and do one short slide of advertisement, because as I said, we are still a young company and we are still hiring. So if you're interested in fighting infectious diseases, BayViro is definitely the place to go to work. And specifically, I'm currently looking for a research associate in our virology department working on hepatitis B virus. So if you're interested, or if you know someone who is interested, please come talk to me after the talk, or go to our website at biro.bio and apply online. And now going back to the acknowledgement slide, thank you very much, and I'm open for questions.
Okay, great talk, and we have lots of questions. You want to start here, Scott?
Yeah, that was great. I'm really interested for the prenylation inhibitor with the mislocalized large antigen. Is it still functional, or is it going to be modified somewhat differently and not even used in replication now?
Right, it's not prenylated anymore.
Yeah.
Therefore, it cannot assemble into a mature virus that can be secreted, so it's just accumulated. However, whether that function, so usually the small delta enhances replication, and then later in the viral life cycle, the large delta is expressed, which dampens down viral replication but starts the assembly and release process. And in that case, we are not sure whether this unprenylated large delta antigen can inhibit it, or whether then the replication is basically increased in the cells.
I guess I'm wondering, does the mislocalization correlate with a lack of prenylation? Right, so is everything that's localized correctly prenylated correctly, and everything that's mislocalized not prenylated?
That is a great question. We don't know that, yeah. Okay. Also, I'm not sure how to test for that. We would have need a specific prenylation dependent antibody. Yeah, that would be great, yeah.
Thank you.
Given your grid of the interactions between the different genotypes of delta and hepatitis B, and knowing the sequences of the delta antigen and the surface protein, can you actually make predictions as to which sequences would be more likely to facilitate replication? And furthermore, can you make sequences about the part of the protein, the three-dimensional structure that's involved in the interaction?
Yeah, so we haven't done that, but that is a great idea, yeah. Because now we really can narrow down and do this experimentally, not just by modeling, to see which associate best, and then to see whether, because that's also part of the large delta antigen that assembles with the HBs antigen, so if we have a closer look at this sequence, we might be able to predict exactly that, yeah. Thanks.
So I have a question on the clinical front. So there's no therapy for hepatitis delta virus, but if you treat the underlying hepatitis B, does that control the HDV replication, or does it still keep replicating?
So the treatment for HPV is just as limited as for Delta, right? So we can treat with nucleoside analogs, which completely abrogates the levels of HPV virions, but it does not at all affect intracellular replication. Basically, Delta just needs the HBs antigen, which is still present. So with the current therapeutics for HPV, we cannot get rid of HBs antigen. And as long as that is still present, Delta will just replicate fine.